Hello and welcome back to Pharmacology Simplified. In today's video, we are going to discuss about hemostasis. Hemostasis is a process that is aimed towards limiting the blood loss after an injury. So basically, we can say that the cessation of blood flow can also be referred to as hemostasis, but after an injury, not in the physiological condition. The term hemostasis, it should not be confused with homeostasis, because homeostasis is a condition where our body continuously carries out several reactions in order to maintain a constant internal environment. So let us see that whenever there is an injury, there are three stages that take place. The first step is transient vasospasm, second step is platelet plug formation, and the third is the clotting. So see what happens, that let us say that this is a blood vessel, and in this blood vessel the innermost here we have as the endothelium. This is the endothelium, right? So if there is an injury, what will happen, that the integrity of this will be lost, and what will happen, that the blood that is flowing, it will start to go out, and the body's approach is to prevent blood loss. So what happens, the very first thing is that there is transient vasospasm, which means that in this there are vascular smooth muscle cells, right? These are vascular smooth muscle cells. What these smooth muscles do, that they regulate the caliber of these blood vessels, right? Like they can increase or reduce the lumen of the blood vessel and limiting or enhancing the blood flow to the target area. So first thing that happens is the transient vasospasm. So there would be contraction in the vascular smooth muscles. As a result, there would be vasoconstriction. If there is vasoconstriction, so let us assume that this blood vessel with an initial lumen of this lumen will now be reduced to this. If the lumen is reduced, this will lead to reduced blood flow, right? There would be reduced blood flow, and after this there is second stage that is called as platelet plug formation. So the platelet plug formation is a process where the circulating platelets in the blood, right? I'm not talking about the other cells, the WBCs, the RBCs. I'm only focusing on the platelets. So for example, these are your platelets, right? These are platelets that are circulating. So whenever there is an injury, on exposure to the negatively charged collagen, the collagen that is present here, right? This negative charged collagen, what happens that, and there are certain factors that are also released from the endothelium which activate these platelets. So what is the activation of platelet means, that these platelets start to change their shapes. The shape of these platelets start to change, and from this shape they tend to become like this. They start to get some projections, right? And there are certain factors. What those factors do, that they help anchor these platelets together. So what happens, that there forms a mesh of these platelets, right? So this platelet formation, what happens, this is platelet adhesion. The platelets, they become sticky, they start to adhere onto the surface, right? And they form a plug. Like we can see, like if this is a wash basin, you are looking at it from the top, and there is the drain hole. So if you put something here, then the water will not go out of this. So some kind of, it is analogous to this only, that these platelets, what they will do, they will form a plug here, and when the plug will be formed here, then the blood will stop flowing, like, or the flow of the blood would be reduced. The third process is actually the most important one, and we call it as blood clotting, right? So blood clotting is a process where the actual clotting of the blood starts to take place, right? So blood clotting, there are clotting factors, right? There are clotting factors in the blood. What are these? These are certain proteins, right? These are certain proteins, and we have 13 clotting factors. Out of these, the factor 6 is still missing. We do not know. So there are total 13 clotting factors, and these are some proteins which are inactive in the normal physiological conditions. But whenever there is an injury into the in the blood vessels, these clotting factors, they get activated, and then they carry out a complete cascade of the clotting process, which I would be explaining now. So this is blood clotting, and before we move ahead, there are a few terms that we should know. Clot of the blood is also called as thrombus, right? For example, this is a blood vessel and it was injured at this site, and here is a clot. So we will call it thrombus. We will call it thrombus. But if because of certain reasons this clot leaves its place, right, and starts moving in the circulation, now it is called as an embolus. So embolus is basically a disrupted version of the thrombus. So we can also see that this embolus is like more dangerous than this thrombus, because thrombus is localized. It may or may not be restricting the blood flow to a vital organ, but this embolus, it is free. It can block, it can go and can block any fine blood vessel which may be like sending the blood to a vital organ, correct? So these are two terms, thrombus and embolus. Right. So if we see here that what I have mentioned here are the various clotting factors that we have. So in total we have 13 clotting factors, out of which the factor number 6 is missing. We do not know about the factor 6, but it is there. So there are the factors. The factor 1 is fibrinogen, second is prothrombin, third is tissue factor. This is an important one. Then we have calcium ions, then we have factor 5 proacceleratin, factor 7 proconvertin, factor 8 antihemophilic factor A. Then we have factor 9 Christmas factor, factor 10 is a Stewart factor, factor 11 is antihemophilic factor C, factor 12 is Hageman factor, and factor 13 is FSF fibrin stabilizing factor. Now we will like move ahead to see the clotting cascade, and we will be like encountering all of these clotting factors one by one. See, these 4 clotting factors, factor 2, factor 7, factor 9 and factor 10, these 4 factors, they are dependent on vitamin K for their synthesis, right? Without vitamin K, these factors cannot be synthesized. So we see here that the clotting pathways, they are categorized into 2 major classes, the extrinsic pathway and the intrinsic pathway. So the extrinsic pathway is like it is dependent on any external injury that may disrupt the vasculature, or there is any damage to the tissue. And this is a rapid process, like it starts within a few seconds of the injury, and it requires external tissue trauma, as I just mentioned, and it starts with factor 3, right? It starts with factor 3 called as the tissue factor, as I just mentioned, that the factor 3 is actually responsible for the initiation of this extrinsic pathway, while your intrinsic pathway is a slow process and it does not require any external tissue damage, right? Only a damage to the endothelium will trigger the intrinsic pathway. Now we will understand and see each and every step of the clotting process. So as I have just mentioned, that the clotting process is completed like by two pathways. One is the extrinsic pathway or the rapid pathway, and other one is the intrinsic pathway or the slow pathway. And all the 13 factors, 13 clotting factors, missing the sixth one, are involved in this. So now we will go ahead with one by one of these understanding of these pathways. So your extrinsic pathway, right? It starts with any injury to the tissues, right? Whenever there is an injury to a tissue, then this extrinsic pathway will be initiated, and this is a rapid pathway, mind you. So whenever there is any damage to the tissue, this will lead to the release of clotting factor 3, that is tissue factor. If you may refer to the previous list also, that there is release of factor 3. Now this factor 3 gets activated, and see, I must tell you that these clotting factors, they are present in the blood all the time, right? Some are being like continuously synthesized, or some are like having long half-lives. We are not getting into that, but all of these clotting factors are present inside the blood, right? But whenever there is an injury, then activation of one factor will lead to this clotting cascade, right? So whenever there is an injury to the tissue releases factor 3, and this factor 3 activates this factor 7. This factor 7 is activated, so this is the inactive form, and A stands for the activated factor. So this factor 3, upon that is released by the injury to the tissue, so this factor 3 will now activate this factor 7 to 7A. This is activated. So now this factor 7A, under the influence of calcium, this is a clotting factor 4, and phospholipids, right? They will activate this factor 10, right? This factor 10 now will be activated in factor 10A, right? So this is the extrinsic pathway. See, the steps are less and this pathway is rapid. The intrinsic pathway is, however, the slower one, more complex. So whenever we are like not talking more about the extrinsic pathway, we will now take it into consideration when we reach to the common pathway. So this is the intrinsic pathway. So intrinsic pathway starts from damage to the endothelium, and endothelium is the innermost lining of the blood vessels, right? That helps the blood vessel in relaxing and contracting. So whenever there is damage to the endothelium, right, there is activation of factor 12 to factor 12A, right? This factor 12 is called as the contact factor. Like whenever your blood cells, right, whenever this factor will come in contact with any other surface than the endothelium, it gets activated, right? So this is the reason whenever we collect the blood into the in the glass vials, the blood gets the blood starts to clot. So this factor 12 gets activated into factor 12A, and this factor 12A now activates this factor 11, which is in inactive form at this point of time, but it is now inactive. It is activated into factor 11A. Now this factor 11A, under the presence of factor 7A, right? Under the presence of factor 7A from the extrinsic pathway, see, these two pathways are like not separable, right? They are like parallel pathways. Whenever we cannot say that if there is a damage to the tissue, then there would be no damage to the endothelium. If there is any damage to the endothelium, there would be no damage to the tissue. So both of these events will occur simultaneously, and both of the pathways will start to like proceed. So this factor 7A from the extrinsic pathway, now this factor 7A and calcium along with factor 9A, factor 11A will convert this factor 9 into 9A, right? Now this factor 9 is activated. This factor 9 is this factor 9 activated, factor 9A, what is it is now doing? It is converting this factor 10 into 10A. Like factor 10 is present in the blood, so it can be activated either by factor 9A or either by factor 7A. Now you must be thinking about why this factor 8A is not being described here. It will be described here after we reach here, right? So from the common path, from the extrinsic pathway and from the intrinsic pathway, we have reached a common destination, that is the activation of factor 10, where it has been denoted as factor 10A. So now this factor 10A, right? This factor 10A, it combines with factor 5, right? It combines with factor 5A, phospholipids and calcium, and it produces thromboplastin, right? It produces thromboplastin. Now this thromboplastin is in presence of calcium. Calcium is also present in the blood. Now this is interesting. Now this thromboplastin in presence of the calcium is going to act on prothrombin, right? This thromboplastin is also known as prothrombinase. So this thromboplastin will now act on this prothrombin. This is factor number 2, refer to the list. So this prothrombin, factor 2, now this will be activated by thromboplastin in the presence of calcium into thrombin. Thrombin is factor 2, right? So this prothrombin inactive is now converted to thrombin, factor 2 activated. Now this thrombin, it is fueling 3 other pathways. It is activating this factor 8 into factor 8A. So this process will now become even more rapid, right? So this is why like I was not describing this factor 8 here. So this thrombin factor 2A will now activate this factor 8 into factor 8A, which will further fuel this conversion of factor 10 into 10A, right? Along with this thrombin also hastens the process of factor 5A, phospholipids and calcium to synthesize more and more thromboplastin, so that more and more prothrombin will be converted into thrombin, and the clotting process will become more rapid. Along with this thrombin also activates your platelets, and the platelets start to adhere. There is platelet adhesion under the influence of factor 2A, and this platelet adhesion will also lead to synthesis of phospholipids from the membrane, because the platelets will become activated, the membranes will start to break down, and these phospholipids, they are like used in many places, like here they are used, here they are used, right? So these phospholipids will be used in the clotting process. Now this thrombin, right? This thrombin, what it will do, like we have fibrinogen, like fibrinogen was clotting factor 1. This fibrinogen is clotting factor 1, but it is inactive, in an inactive state. So what happens, this fibrinogen will be this fibrinogen is soluble. This is in soluble form in the blood. So this fibrinogen, which is in soluble form in blood, will be acted upon by this thrombin, and it will be converted into fibrin, which is insoluble, correct? Which is insoluble, and there will be starting formation of fibrin threads. And then this thrombin will also activate your factor 13. Factor 13 was your FSF, fibrin stabilizing factor. So name is clearly showing that fibrin stabilizing factor, it is a factor that will be stabilizing your fibrin threads. So now this factor 13 will be activated by thrombin into factor 13A, and this factor 13A will be acting on fibrin, and this fibrin, which is till point it was insoluble, but after the action of factor 13A, it will become stable fibrin thread, right? It will become stable fibrin threads, correct? So this is how the clot will form. So we can see that here we have the extrinsic pathway, then we have the intrinsic pathway, then the reactions occur, then we have the common pathway, and after the end of the common pathway is the formation of fibrin threads, right? So this is one thing, like how the clot is forming. But after the repair process is complete, or when the repair process is going on, the clot itself starts to dissolve, right? We do not have to do some conscious efforts in order to get the clot dissolved. So what happens that there is plasminogen, right? Plasminogen, it is present, plasminogen is also present in the blood. So it is like circulating, and this plasminogen, it is converted into plasmin, right? This plasminogen is converted into plasmin, and this plasmin acts at two sides. First, it will act on the fibrin, the stable fibrin, in order to break down that clot that has been already formed, right? So the clot that has been formed by all this process, the fibrin, so it will start to break down these fibrin threads, right? This stable fibrin will be broken down into smaller fragments, and along with this, what will happen that this plasmin will also act on the fibrinogen, right? Although this is inactive, but what this process is doing, that it is reducing the availability of the substrate for thrombin in order to make fibrin threads. So this plasmin will act on the fibrinogen, and this fibrinogen will be degraded into smaller fibrinogen breakdown products. Correct? One more thing, that the activation of plasminogen into plasmin takes place by tissue plasminogen activator, and it is released from endothelium, right? So this plasminogen is converted to plasmin by the tissue plasminogen activator, and which is released from the endothelium, right? So endothelium, when it starts to heal, it also starts to release this TPA, tissue plasminogen activator, which like start the process of fibrinolysis. This process is called as fibrinolysis. If we say that this is the extrinsic pathway, this is the intrinsic pathway, this is the, like this part is the common pathway, and this part is the fibrinolytic system, fibrinolysis we will say it, right? And this is fibrinolysis. So this is how the clotting process takes place. So with this, I would put an end to this video. And if you have been able to understand anything out of this video, please make sure to subscribe to the channel, like the video, share the video, and happy learning to all of us. Thank you very much.